Chapter 24, Glycogen Degradation. Glycogen is a very large branched polymer of glucose residues. Most of the glucose residues in glycogen are linked by alpha-1,4 glycosidic bonds, and branches at about every 12th residue are created by alpha-1,6 glycosidic bonds. Glycogen is not as reduced as fatty acids, and consequently not as energy rich. However, the readily mobilized glucose from glycogen is a good source of energy for certain strenuous activity. Unlike fatty acids, the released glucose can provide energy in the absence of oxygen and can supply energy for anaerobic activities. Moreover, the controlled release of glucose from glycogen maintains the blood glucose concentration between meals. The circulating blood keeps the brain supplied with glucose, which is virtually the only fuel used by the brain except during prolonged starvation. Let's look at the big picture again. Where's this chapter? Is glycogen to glucose. Chapter outline. First, we're going to study glycogen breakdown requires several enzymes. Then phosphorylase is regulated by allosteric interactions and reversible phosphorylation. Epinephrine and glucagon signals the need for glycogen breakdown. Glycogen is a highly branched homopolymer of glucose present in all tissues. The largest storage of glycogen are in liver and muscle. The liver glycogen breaks down and releases glucose to the blood to provide energy for the brain and red blood cells. In the liver, glycogen synthesis and degradation are regulated to maintain the concentration of glucose in the blood required to meet the needs for the organism as a whole. Muscle glycogen are mobilized to provide energy for muscle contraction. Let's look at the glycogen structure. A. The glucose unit joined by alpha-1,6 linkage are shown as straight lines. The non-reducing ends of the glycogen molecule form the surface of the glycogen granule. At the core of the glycogen molecule is the protein glycogenin, yellow. Degradation takes place at this surface. B. A cross-section of a glycogen molecule. The glycogenin is identified as G. Glycogen breakdowns require several enzymes. The glycogen to break down to glucose 6-phosphate require 4 enzymes. First is a phosphorylase, cleaves glycogen to release glucose 1-phosphate. Then a transferase shifts a small oligosaccharide near the branch point to a nearby chain, making the glucose moieties accessible to the phosphorylase. Then a debranching enzyme, alpha-1,6 glucosidase, cleaves the alpha-1,6 bond at the branch point, releasing a free glucose. Then the phosphoglucomutase forms glucose 6-phosphate from glucose 1-phosphate with the use of a glucose 1,6-bisphosphate intermediate. Let's look at the first step. The phosphorylase cleaves glycogen to release glucose 1-phosphate. Glycogen phosphorylase degrades glycogen from the non-reducing ends of the glycogen molecule catalyze a phosphorylases reaction that yields glucose 1-phosphate. Glucose 1-phosphate is later converted to glucose 6-phosphate by phosphoglucomutase. Let's look at the reaction. The glycogen with N residue break down at the non-reducing end, form a glucose 1-phosphate plus a glycogen with N-1 residues. So the phosphate, the orthophosphate, is added to the 1,4 glycolysic bond and break the bond. This reaction is called phosphorylasis. Think about the hydrolysis is to add water to break the bond, and phosphorylasis is to break a bond by adding of orthophosphate. Here's the reaction. The glycogen plus Pi produce 1 glucose 1-phosphate and a glycogen with 1 less residues. Let's look at the glycogen structure. What is a non-reducing end? Non-reducing end is the end of a branch with a free OH group on the carbon. The two non-reducing end is marked as red with the arrow point to it, and the beginning of the branch is marked as green. It linked by a alpha 1,6 linkage. And we also see that alpha 1,4 linkage on the figure. A deep branching enzyme is also needed for break down the glycogen. Glycogen phosphorylase can only cleave alpha 1,4 glycosidic bonds and stops when it reaches a residue 4 residues away from a branch point. A transferase shifts a block of 3 glucosyl residues from one outer branch to a nearby chain so that the phosphorylase can continue the degradation. A deep branching enzyme called alpha 1,6 glucosidase then cleaves the alpha 1,6 bond at the branch point, releasing a free glucose. Let's see how this glycogen molecule being broken down. This glycogen molecule has one branch linked by alpha 1,6 linkage. The rest is linked by alpha 1,4 linkage. Catalyzed by phosphorylase, then it goes the phosphorylases reaction, adding orthophosphate to the non-reducing end and release a glucose 1-phosphate. This process keeps repeating until it reaches 4 residue away from the branch point. Then the transferase remove the 3 block of glucosyl to link to a nearby chain so that the phosphorylase can continue catalyze the phosphorylases reaction. After the transfer, there's one glucosyl residue is exposed with alpha 1,6 glucosidase undergoes the hydrolysis reaction. This glucose marked as a green color being released. Let's compare how 1,6 glucosidase work versus phosphorylase. From last slide, we saw at the point when there's only one glucosylate left, linked by alpha-1,6 linkage with the glycogen. At this time, it will undergo hydrolysis reaction catalyzed by alpha-1,6 glucosidase, release one glucose, leave the glycogen has one less residues. So alpha-1,6 glucosidase will break down the alpha-1,6 linkage and release one glucose. Well, we saw this reaction before, that with to alpha-1,6 linkage, the enzyme is phosphorylase to break down at the non-reducing end, release one gluco-1-phosphate, and the glycogen have one less residue. So the difference between 1,6 glycosidase and phosphorylase is alpha-1,6 glucosidase will break down alpha-1,6 linkage, release a glucose. Is a hydrolysis reaction. With phosphorylase, it breaks down at the non-reducing end, release the gluco-1-phosphate, not the glucose, it's gluco-1-phosphate. And this reaction is called phosphorylases reaction. Last step, the glycogen release gluco-1-phosphate. However, gluco-1-phosphate has to transfer to glucose-6-phosphate in order to enter the gluconeogenesis pathway to convert to glucose. The enzyme catalyzes this reaction is phosphoglucomutase. Through this enzyme, a phosphor group is transferred from the enzyme to the substrate, and a different phosphor group is transferred back to the enzyme to its initial state. Once the glucose-6-phosphate is being produced, it needs glucose-6-phosphatase to turn glucophosphate into glucose through gluconeogenesis pathway. The glucose-6-phosphatase generates free glucose from glucose-6-phosphate in liver. The free glucose is released into the blood for use by other tissues such as brain and red blood cells. Glucose-6-phosphatase is not present in other tissue, including the muscle. Glucose-6-phosphate catalyzed by glucose-6-phosphatase enzyme, it will produce the glucose plus a orthophosphate. That's the last step in gluconeogenesis. Phosphorylase is regulated by allosteric interactions and reversible phosphorylation. The key regulatory enzyme for glycogen degradation is glycogen phosphorylase. Phosphorylase exists in two forms, a less active B form and a more active A form. The A form differs from the B form in that a serine residue is phosphorylated. Both the A form and the B form display RT state equilibrium. In the B form, the T state is favored, while in the A form, the R state is favored. Recall that T state is tension state, is more interaction, more stable, lower affinity for oxygen, while the R state is relaxed state, has a fewer interactions, more flexible, higher affinity for oxygen. A key role of the liver is to maintain adequate blood levels of glucose. As a result, the default state of liver phosphorylase is the A form in the R state. From the previous slide, we learned that the A form is more active and the R state is relaxed state, which has a higher oxygen affinity, which means that the liver phosphorylase is prepared to generate blood glucose unless signaled otherwise. Glucose is a negative regulator of liver phosphorylase, facilitating the transition from the R state to the T state, which is easy to understand because the liver intends to maintain a stable level of glucose. When the glucose level is high, then the liver phosphorylase will be inhibited to produce more glucose, which facilitates through change from R state to T state, which is a lower affinity to oxygen and less active. Liver phosphorylase and muscle phosphorylase are isoenzymes. Isozymes or isoenzymes are enzymes that are encoded by different genes yet catalyze the same reaction. Let's look at the phosphorylase regulation in structural detail. The right side is the A form, the left side is B form. Active sites are marked as a red circle inside the enzyme. And you can see the A form different from the B form is that the two phosphorylation attached to the regulatory structure. The bottom is the T state. The A form and B form, they both have this T state where the regulatory structure, the blue ribbon and the green ribbon, stick into the active site, bound to the active site, partially block the active site, make it less active. For the A form, it's more active because the equilibrium between the R and T favors to the R. You can see the big arrow towards to the R. While the B form, the equilibrium favors the T state, which is tense state and less active. This figure shows the allosteric regulation of liver phosphorylase. The bonding of glucose to phosphorylase A shifts the equilibrium to the T side, shows it to the left, and inactive the enzyme. You can see the glucose is bonded in the active site inside the phosphorylase, which causes the equilibrium to the T state, which inactivates the enzyme. Thus, glycogen is not mobilized when glucose is already abundant. In muscle, the muscle phosphorylase is regulated by the intracellular energy charge. In the liver, we learned that the phosphorylase is regulated by the glucose level, and the muscle is regulated by the ATP level and AMP level. In muscle, the default form of the phosphorylase is the B form in T state. The B form is the inactive form, and T state is tense state that it has a low affinity to oxygen. When energy is needed, as signaled by an increase in the concentration of AMP, the phosphorylase bonds AMP, which stables the R state. The T state of the phosphorylase is stabilized by ATP and glucose 6-phosphate. This figure shows allosteric regulation of muscle phosphorylase. During workout, a low energy charge state, represented by high concentration of AMP, favors the transition to the R state. When resting, that is a high energy charge, which the ATP concentration is high, then it favors the T state. Skeletal muscle is composed of several fiber types. Type 1, or slow switch fibers, power endurance activities, rely primarily on cellular respiration as a means of generating ATP. Glycogen is not an important fuel for type 1 fibers. Therefore, the amount of glycogen phosphorylase in type 1 fiber is low. Type 2B, or fast switch fibers, use glycogen as their main fuel. Glycogen phosphorylase are abundant in this type of fiber. These fibers are also rich in glycolytic enzymes, which are needed to process glycogen quickly as in the absence of oxygen. Type 2B fibers power burst activities such as sprinting and weightlifting. Type 2A fibers have biochemical characteristic intermediate between the other fiber types. This table shows the biochemical characteristic of muscle fiber types. We can see type 1 in this column, type 1 column. The mitochondrial density is high, and it has high triacetylglycerol content, while in type 2A, it's intermediate in almost every category. Type 2B is high in glycogen content and high in glycogen phosphorylase activity and phosphorufructokinase activity. Phosphorylation promotes the conversion of phosphorylates B to phosphorylates A. We know the phosphorylase A form is active form. Therefore, we can say phosphorylation activates the phosphorylase enzyme, stimulate the glycogen breakdown. In both liver and muscle, phosphorylation of a single serine residue in each subunit converts phosphorylase B into phosphorylase A. This conversion is initiated by hormones. The secretion of glucagon and epinephrine result in the phosphorylation of the conversion. The glucagon secreted when the glucose level is low, like in fasting or starving situation, and the epinephrine secreted when feeling fear or excitement. This regulatory enzyme phosphorylase kinase catalyzes this covalent modification. The enzyme responsible for the conversion of glycogen phosphorylation from unphosphorylated B state to the phosphorylated active A state is phosphorylase kinase. The phosphorylase kinase itself is activated by both phosphorylation and calcium bonding. The phosphorylase kinase is maximally active when phosphorylated and bonded to calcium. The inactive phosphorylase kinase has 4 subunits. The calcium ion bound to the delta subunit become partially active and further activated by pKa hormones and become fully active. This fully active phosphorylase kinase catalyzes the phosphorylase B to phosphorylase A. The core in glycogen is the G proteins. It transmits the signal for the initiation of a glycogen breakdown. Glucagon secreted in liver and the epinephrine in the muscle initiate the G protein cascade that result in the production of cyclic AMP. Calcium, released in muscle to stimulate contraction, initiate the activation of phosphorylase kinase. In liver, calcium release is stimulated by epinephrine binding to the alpha adrenergic receptor, which activates G protein, instigating the phosphorinositide cascade. Cyclic AMP activates protein kinase A with phosphorylate and completes the activation of phosphorylase kinase. Phosphorylase kinase converts glycogen phosphorylase B to the A form, activating glycogen degradation. This figure illustrates the hormonal control of glycogen breakdown. The left side of the illustration shows the hormonal response to fasting. Glucagon stimulates glycogen breakdown in the liver when blood glucose is low. The right side of the illustration shows the hormonal response to exercise. Epinephrine enhances glycogen breakdown in muscle and the liver to provide fuel for muscle contraction. This figure also shows the interaction of different pathways. We can see in the liver, when glucose turns to glycogen, that's the pathway, glycogen breakdown pathway, which is chapter 24, and the lactate turn to glucose through gluconeogenesis, chapter 17. And in the muscle cell, during the exercise, the glucose turn to glucose 6-phosphate, and the glycogen turn to glucose 6-phosphate through the glycogen breakdown, and glucose 6-phosphate turn to pyruvate through glycolysis. And pyruvate, when there's no oxygen present, it turns to lactate, or when intense exercise, it turns to lactate. When oxygen is present, it turns to oxidized to carbon dioxide through the citric acid cycle and oxidative phosphorylation, chapter 18, 19, 20, 21. Glycogen breakdown must be rapidly turned off when necessary. This is easy to understand. When the body doesn't need extra glucose, it should not keep breaking down glycogen. It should keep the reserve. The glycogen degradation is turned off by several mechanisms. First is through the inherent GTPase or through the phosphordiesterase converts the CAMP into AMP, which does not stimulate the protein kinase A, or through the protein phosphatase 1. It removes the phosphoryl groups from phosphorylase kinase and glycogen phosphorylase, therefore inactivated the enzyme. Biological insight. The glycogen depletion coincides with the onset of fatigue. The onset of fatigue coincides with the depletion of glycogen reserves. However, it's not clear whether the glycogen depletion is an actual cause of the fatigue.